I'm really excited to be here today to present this new MaxWell Biosystems assay. In particular, we have already seen two speakers showing very exciting results using this technique to measure axonal signals on top of high-resolution microelectrode arrays. And following up of my talk, we will have Professor Bastian Hengel who will give us a little bit of an insight how this can be used in the pharmaceutical industry. So I'm very happy to present this to you today. Basically, we are with the Axon tracking assay, we have a method to record and to automatically detect and identify propagating action potentials along axons on top of high-resolution microelectrode arrays at high resolution and high throughput. So first, I would like to start questioning why should we measure axonal signals at all? We have seen already a lot of insights today, so very quickly. They represent a new novel functional readout, which allow us to maybe look at mechanisms or effects which were not possible to investigate in that detail before. Furthermore, they're also a biomarker for maturity and developmental stage of the neurons, so they can be used in addition and complementary to the biomarkers that are already established. Of course, they're highly relevant for neurological diseases. So just as one example, in Parkinson's disease was found to cause reduced axonal growth and also axonal degeneration, but also very relevant for a variety of diseases. And finally, as we have also seen in the talk of Dr. Shimba, that the axonal conduction is also thought to play an important role in computation, neural computation, and plasticity. So maybe by looking closer at the axons, we will actually be able to better understand the brain. So as the next question, what type of techniques do we have to record axonal signals? So here on the top, you can see, doesn't work. No, wrong button, sorry. So on the top, you can see the recording axonal signals with whole cell patch clamp technique. This is very complex to do because you're patching the cell soma with one patch pipette and then the axon at different sites with a different patch pipette. So as I said, this is a very challenging technique to master on one hand, and also the throughput is very low because you can record typically one cell at a time and only individual spots along the axon. On the other hand, on the bottom, we see optical techniques such as genetically encoded voltage indicators. So this type of technique, of course, has a very high spatial resolution. On the other hand, has some challenges to overcome, for example, effects on the cells, toxicity, photobleaching, and typically a low signal-to-noise ratio. So the method that we see in the center, which is recording axonal signals with high-resolution microelectrode array, obviously is what we are working with at MaxWell Biosystems. But just to, I think I would like to add one point in comparison to the other techniques. I think maybe the most important reason why we should use microelectrode arrays is because they're really simple to use. All you have to do is to plate your cells on the microelectrode array, and then you can record this type of signals. So just a quick looking back, what was already done with this type of technology. I just chose out of many papers, I chose to show here two. One work in 2013 by our former colleague Douglas Packham, who I think for the first time showed that you can trigger individual action potentials by electrical stimulation, by stimulating at particular electrodes, also correlated it with a staining of the neuron, and showed that he could follow the propagation. On the other hand, we have a paper from 2019 by Thorsten Buhlmann, who basically showed that it is possible to reconstruct the complete axonal arbor, and not only for individual single cells, but also within the same experiment for many different cells. So really getting towards the high throughput. So now I would like to take a closer look about what type of signals are we actually recording with the microelectrode array. If we have here the, let me just, okay, doesn't work. So if we have the representation of a standard neuron, we have the somatodendritic area in the top. We have the axon initial segment with a high concentration of ion channels. And then we have the axons, axon as shown here, or many times we have also multiple axons which propagate the action potential to the other cells. The signals we can record typically look something like that. We have large action potentials around the axon initial segment, and along the axons we have quite small amplitude signals. However, they show a very clear temporal propagation pattern. So taking this together, I would like to highlight why high-resolution microelectrode arrays are a good or an ideal technology to measure these type of signals. First of all, what you need is to have a high spatial temporal resolution, which means that you can record the axon at many different spots, and of course at high temporal resolution. Another very important thing, you need to have low noise readouts, because as I shown in the last slide, the signals are very small. So you need to do averaging, but still the quality of your recording is very important. And if you have a large sensor area, it's very nice because you can really follow the propagation of the action potential over longer distances. So in summary, a system, as you can see here, the MaxTwo, which has multi-well high-resolution microelectrode arrays, allows you to record from multiple neurons and multiple axons on multiple wells simultaneously. And since this is a non-invasive measurement, you can even track and record the same activity over multiple days. Now we are ready to get into the showing the assay itself. I would like to quickly mention the other two assays because basically they follow one after the other. So typically we start an experiment with the activity scan. The activity scan tells us where on the microelectrode array area the cells are located and how active they are. Then, once we have identified the neurons, we can go to the network level. So we select electrode underneath all our cells, or many of our cells, and we record them, all of them simultaneously, which gives us information about the synchronicity of the network and about bursting patterns, as you can see here in the slide. And finally, with the axon checking assay, we go into the single cell level. So the results that we obtain from this assay are really on a single cell basis and even give us access to sub-cellular features such as the axonal processes. Now, I would like to introduce you to the general framework of how what is involved if you do such an experiment. First, we always have the assay part, which is the recording. Then, once you have recorded the data, you can run the analysis. And finally, you have metrics that are calculated, which you can export to run your individual statistical analysis. So just quickly looking at the assay part, so how to record this type of signals, we start by doing an activity scan. So we have identified the cells on the array. Then we select what we call neuronal units, or these fixed electrodes, which are basically the location of the putative neurons we want to resolve. And then we also define a scanning area. So we will try to reconstruct the electrical propagation of these selected neurons on the entire scanning area. Usually we take the entire array, but we can also limit that if we want. Now, once we have recorded the data, we go to the analysis step, which is composed out of 3 components. In a first part, we need to spike sort the data. So what does it mean, spike sorting? Out of all the spikes that we record, we want to identify ones that are belonging to one single neuron. Typically, and out of this spike sorting step, we take the most prominent cluster. We use the spike times of this selected cluster to compute what we call the footprint of the cell, which is basically the average extracellular signal of these particular neurons. It means that whenever this neuron fires an action potential, it looks something like that, if we look just at the amplitude representation. And then the last step of the analysis for the axon tracking assay is basically the core, is an automatic tracking algorithm which follows the signals of propagating action potentials and reconstructs what we can see on the bottom, the axonal arbor of this particular cell. All right. Once we are there, we can really start looking at the metrics. So what kind of information can we extract from it in order to characterize our cell lines, our neurons? Of course, we have the neuronal conduction velocity, which is a weighted average of the individual conduction velocities of the branches. So you also have access to the individual branch velocities or to this average conduction velocity. We extract the amplitude at the initiation site. We also extract the length of the longest branch. Another metric which is interesting, which you see on top, is for how long do the axonal signals travel for this particular neuron. It's also kind of a measure of the maturation. So basically, we look at the latest axonal signal, which is still associated with that neuron. If we sum up all the individual branch lengths, we come up with a total axon length, which is, of course, also an important metric. And finally, we're also interested in the outreach of the axons of the particular neurons, meaning do they really go far away or do they stay all within close of the center? And we measure that by using the longest distance from initiation site, basically just the direct measure for this. So with that, I would like to show you some examples how it looks in practice. So here are some examples of individual axon tracking. This is from iPSC-derived glutamatergic neurons. And I have to say that these were done after 63 days in vitro. That's why they show this very long propagation. So they have grown on the chips for a long time. What you can see in the back, basically in blue, is the movie of the propagating action potential, and it's really just the signal that we extract from footprint extractions. It's not processed, just bandpass filtered. And then on top, the red lines are showing the reconstruction of the axonal branches, which are done by the software. So we get asked many times if this is a problem if we have dense cultures, what happens if the axons overlap, can they still be resolved? And the answer is yes, they can be resolved. Even if you have a dense culture which has a lot of activity, is even good for the assay, because you need a certain level of spontaneous activity. And since we are averaging based on the spike times, we really can average out any overlaps. So as you can see on the figure here in the bottom, we get a lot of neurites from this particular experiment. So we can quantify all these neurites, but we can also look at them for every cell individually. So meaning that for every cell, we extract all these metrics that we have shown before, and we can resolve them. Maybe one addition to this particular slide, of course, if you compare it with standard optical imaging techniques, you see the advantage of this method, because if you use optical imaging, many times it's difficult to take apart the processes or the neurites from different cells. Of course, depend what type of method that you use. So I would like to wrap up with one last comparison that I want to show you that was done in collaboration with the bioengineering laboratory of ETH Zurich. Here we use the axon tracking assay to record from human dopaminergic neurons, from human glutamatergic neurons, and human motor neurons, all iPSC-derived human cell lines, of course. And we were able to detect single cells for all these different cell types. And interestingly, we found a significantly higher conduction velocity, axonal action propagation conduction velocity for the motor neurons as compared with the dopaminergic and glutamatergic neuron. So before I summarize my talk, I would also like to take the opportunity to thank everybody that was involved here. Basically, all the team is here, so we can have some follow-up discussions, but I would like to mention Blondine, who is also here, who contributed to this a lot to this assay, and then also Anais contributions on the software and all the recordings and help with the data analysis, the software engineers at MaxWell Biosystems, Stefan and Tobias, and also Silke and Elvira from the scientific team for all their help with the cultures on our side. So In order to summarize my talk, we showed that we can record and identify single neurons at high throughput with high-resolution microelectrode arrays and with this new MaxLab Live assay. We showed that we can track the morphology of axonal arbors and that we can extract conduction velocity of action potentials along multiple axons. So thank you very much.
Before starting, I just want to thank MaxWell for inviting me to be here. It is a pleasure, and not only to be here, but also working with them is a really nice crew. I think they have really nice people in the company.
Thank you.
So yeah, today I'm going to be talking about the model that we use and also some applications. I want to focus on the applications that we have together with MaxWell. Just a disclosure, I'm a name co-inventor of patent at Johns Hopkins, and now it is licensed by Axon. So today I'm going to be talking about this model that we use. This was developed through some funding from Human on a Chip. There was a huge funding from DARPA, FDA, and NIH. They put 70 million into this. The idea was to generate microfluidic platform that has different microphysiological systems all interconnected between them, with the idea to use for toxicology purpose, also for drug screening. So most of the money was for MIT and Harvard because they were basically developing the platforms, but there was a small funding for small groups that were developing these microphysiological systems. And we were lucky to get some of this money at Johns Hopkins Bloomberg School of Public Health in collaboration with the Johns Hopkins School of Medicine with Hong Yun Song Lab. And we got this money to try to develop something related with brain. So basically what we did is something similar to what many people have done. We take induced pluripotent stem cells and then we differentiate it to neuroprogenitors. When we had the neuroprogenitors, we put them in a shaker like this. I brought a video. Basically, we put it in a 6-well plate and we put it in this type of shaker. Basically, the shaker, what it does is to generate neurospheres. So neurospheres are basically spheroids that contain neuroprogenitor cells. So when we have them generated, we can generate around 80 spheres in each well. So you see, we can obtain a lot of tissue, what is really convenient when you want to do several experiments. And then when we have it in this particular situation, we add differentiation media and we keep them for up to 8 weeks. So after 8 weeks, we obtain something like this. It's around 350 microns sphere. They are quite homogeneous in size and shape, and they contain different neuronal cell types all together with glial cells. We have studied some synaptic formation also to make sure that they form synapses, as you can see here is in green for synaptophysin marker. But we have also put a lot of effort to try to understand the glial cells. We also have different neural cell types, for example, dopaminergic neurons here in red, but we have also studied and we have also for GABAergic neurons and cholinergic neurons. And we have put also a lot of effort to try to understand if we have glial cells, so because to differentiate into, for example, oligodendrocytes has been really challenging in vitro cultures. So we have put a lot of effort to try to understand if we can get a nice population of oligodendrocytes, and also if they are mature enough to produce myelination. So here are just a few of the markers. But one of the things that we consider more interesting is when we study myelin basic protein, we saw that we have here in green, we saw that we have a lot of myelin, and I will show you a little bit later a little bit more in the characterization. So like I said, we put some effort to characterize the model, but this is something that will continue ongoing, I think, for years. But basically, with the neuropathology department at the Johns Hopkins University, we did several stainings that they normally use for patients to study the different types of neurons, astrocytes and oligodendrocytes. And also, we spent, I don't know if this is working. Yes, oh, nice. And we also did some studies on the myelin. So we saw like the myelin is growing over the weeks of differentiation. And also we did some confocal imagings to make sure that the myelin was wrapping, and also some electron microscopy to make sure that the myelin is actually wrapping in a compact way the axons. And we also now doing some lipidomics to study this myelin. But something that was really important for us, because we were going to use this for toxicology and also for drug screening, we wanted to have a reproducible model. So we spent a lot of time to make sure that our model was quite reproducible. So we did some studies for the size to make sure that every time we do experiment, we had the same size and shape. We also did some flow cytometry to make sure that the populations that we are obtaining in this model is quite reproducible over experiments. And we did also with high content imaging, like studying of the astrocytes or oligodendrocytes amount that we have in our aggregates. Also, because we want to use this for toxicology, we need to have a tissue that doesn't have necrotic centers. And literature has shown that when you have a tissue that is higher than 500 microns, you start to see like necrotic centers. So we try to keep the size smaller than 500 microns to avoid this necrotic center, because if we are going to try some chemicals and see the adverse effects of these chemicals, obviously we don't have to have already some dead tissue in the model. We have used the model for many applications. I just want to show two because I want to focus on the multi-electrode arrays. But basically what we did here, we incorporate a tumor into our brain spheres. So the tumor grows over the time. This is a glioblastoma tumor. And then we use something that's called micromatrix for a company in UK. Basically, we add these brain spheres with the tumors in the plate, and we do different treatments with chemotherapies. And then we're using this spheromatrix micro TMA. We can cut different sections and stain for different stuff. And then we can analyze in-house with the software that we have. Because the tumor is fluorescent, we can also track and quantify how much of the tumor we have after the treatments, and also for different stainings. This is just an example. Here we have doxorubicin. It's a drug it's a chemotherapy drug, and you can see here the tumor. This is going higher concentrations, and you can see how the tumor is disappearing when we increase the size, the concentration. And this is really interesting because here we also have the healthy tissue, so then we can establish what is the treatment, the adequate dose for the treatment. Another of the application before going to the MaxWell is the use for myelin quantification. We have an assay on myelin disruption. This has already been published, so if you're interested, you can go ahead, and there's two publications. But basically what we do is to use the model to quantify myelin, and then we do different treatments for developmental neurotoxicity, compounds that are possible, had the potential to produce developmental neurotoxicity, and then we quantify by this model. And this can be also applied for other diseases that are related with demyelination or myelin disruption. So there's several applications. I'm not going to go all over them, but basically we use this a lot for toxicology. Even we have some publications on human virus, stuff like that, but our lab is mainly focused in toxicology and drug screening. But today I want to show a little bit what we have done in terms of electrophysiology, and I apologize because I'm no expert. I just started really recently with this type of work. But basically also when we start with the characterization, we were also interested in to know if we have our model is electroactive, because this is the most important feature of the brain models. And so what we did is to use this MaxWell Biosystems MaxTwo in the beginning. So the idea is we attach the brain spheres or the brain spheroids on top of the electrodes, and then we measure the spontaneous electrical activity. I know that some people don't like spontaneous electrical activity, but in this case, was what we could do. Unfortunately, when we move this into like screening, we have some issues. And the issues of this is because, as you can see in the picture, maybe it's too small, but if you put some imagination, you can see how some brain spheres are on top of the electrodes, right? And some brain spheres are not on top of any electrode. And then when we measure the plate, we see a huge variation between the different wells. If we go to a raster plot, and here you have the different wells on the right, you can see how there's a lot of variation between one well and the other well. And that was getting us crazy when we want to establish the neurotoxicity of some of the drugs that we were testing. So a lot of years later, I would say 3 years later, when I moved to Switzerland, I got aware of this company, MaxWell, you all know, because our collaborators were using MaxOne. And then we thought that we could use this for our model and actually will help us to reduce the variation between the wells. So because now we can attach the brain spheres into the electrodes, and by using the activity scan, we can go through all the electrodes. Now we can only select the ones that are active, and that will reduce a lot the variation that we had before. And now we can always select around 1,000 electrodes and measure on that. Also because it's quite easy to identify the spheres by the picture. So it was easy to identify, what are the brain spheres that we want to use. So this was a good advantage in reducing some of the standard deviation that we were getting because of the variation between wells. So I want to show you 2 projects that we are working on together with MaxWell, because I don't know how much you guys know about toxicology or regulatory toxicology. I just want to show you a little bit of the background and why we are doing this work. So as you probably know, when chemicals go into the market, right, they need to be safe for human population and also for environment. So they have to go after rich legislation. Now the chemical industry have to make sure that they make a dossier with all the tests necessary for a chemical going to the market, right? So they have to follow some guidelines. There are some tests that they do, and normally these tests are made in animals. Most of them are made in animals. So the problem at the moment is we have many chemicals that have not been tested yet. We have more than 100,000 chemicals in the chemical universe and more than 4 million mixtures. And it has been really difficult to basically for now, because the responsibility is of the chemical industry to get all the money to be able to test all these chemicals. So the main problem is the actual guidelines for the testing is really time consuming and expensive, and there are some ethical concerns of using animals for this. And Switzerland is a really good example of how things are moving to try to reduce the amount of animals. But there is also other acts like in US, and also now OECD is moving through trying to reduce this amount of animals. But obviously there are also some scientific concerns, right? Humans are not the same as animals, and there is in literature many cases where we see some effects in animals that we don't see in humans and the opposite. So a few years ago, there was the National Council of Research. This was experts from all over the globe and also from academia, industry, and regulatory bodies that they wrote this book. So the idea was to try to understand what is the future in toxicology. And this has been named Paradigm Shift in Toxicology, or Toxicology in the 21st Century. And some of the pillars for this change has been focused on high throughput and high content in vitro assays, the use of more human cells, and also to understand the mechanism of toxicity, so we can use weight of evidence to assess all these chemicals. So from that, many people have been trying to develop different assays, especially in US. We have NCAST program and TOX21, that there's a huge program on in vitro assays by using high throughput technologies. One of the assays that they have been using and the responsible of the unit is Timothy Schefter. He is the EPA, Environmental Protection Agency. They are using electrophysiology activity to assess neurotoxicity. So they use this machine, and they use cortical rat neurons to assess the toxicity. Basically, they expose the chemicals for up to 12 days, and they do like repeated dose. And they have screened hundreds of chemicals in the last years, and this has been really useful for the field. However, because as I mentioned before, mice are not human, we thought that maybe we could try, in collaboration with them, to use our model to assess this chemical toxicity. And a few years ago, with MaxWell Biosystems, we applied for a Novartis grant, and we got some money, and also from SCAT, and that's why we are doing this. So basically, in toxicology, when you want to put an assay and to make sure that the assay works, you have to use something called training set. So basically, chemicals that you already know that produce neurotoxicity. And then you want to see, okay, is my assay able to detect this neurotoxicity? And sometimes it's really hard to find the chemicals, but in this case, it was really easy because they were already being tested by Tim Schefter at the EPA, right? So we select these compounds. We have positive controls, and then we have 2 negative controls that we also need. And the idea was to test this in our model. The first thing we have to do in toxicology is to do the cytotoxicity, general cytotoxicity, because in our case, we are really focused on neurotoxicity. So if the cells are dying for other reason, then we cannot say that it's a neurotoxic effect. So we have to find the concentrations, and here I show some of the viability, or maybe not. Yeah, some of the viability assay for different compounds. We saw only that loperamide was toxic at the concentration we were using, and the other ones were not cytotoxic. The range of concentration was easy to find because also, like I said before, we were using already data from EPA. So the experimental design is we do the full activity scan, full resolution, and then we manually select the spheres. And then we apply the compound, and we have different concentrations, and we do this for 13 days. So this is repeated over the time. So basically what we get is something like this. So we have all the spheres marked, and then we see how the electrical activity goes through all these spheres. If we go more in detail, and just to show it closer, you can see the spheres and how they are spiking. So what we are trying to do now is not finished yet, and I think we still have a lot of work to do, and I'm really happy that MaxWell is helping out with this, because I'm not super expert. But what we are doing now is to try to establish the toxicity of these compounds. So here, it's really small, sorry guys, but basically, we had the different times, the different days of exposure and the different concentrations. And now we can start seeing some dose responses. That is what toxicology people like, right? We like those responses. Just to make it easy to you, because I didn't want to put a lot of crazy stuff, Anna did this amazing graph. Thank you very much. I know she worked hard because she sent me this on Saturday. So. And so basically, this is the area under the curve of the mean firing rate of the whole every days. So here you can see how the controls don't do anything, right? There's no statistical significant changes. But when we go to the compounds, we start seeing these dose responses, even at really low concentration for some of the compounds, right? 0.1 micromolar. And we see dieldrin going down, and we see deltametrin going down. We didn't see much in valproic acid. So this is basically what we are trying to do. We still have some issues, and the main issue is because MaxWell analysis is made for 2D, and now they are trying to move to 3D, but obviously it takes some time. We get all the output of the readout from the whole well, right? So we cannot identify the different aggregates. So what we are trying to do now with them, well, basically, they are trying to do, because I don't do anything of this, but basically what they are trying to do now is to find a way that we can analyze each of these brain spheres separately. So for example, here, if you see in red, and the idea will be to extract the data on this specific area. And this is because we are also interested in other things like bursting and things like that, that is really hard to do when you have like different tissue in the same well. So hopefully this will be done soon, and we can start doing the comparison between the data already from EPA. So this is ongoing. Also, I want to show you another small project that we have in collaboration with Mehdi Tafti, especially Moji, that he's the driving force of this project. And again, I'm not really expert in sleep. So, but basically what they are trying is to develop a system for sleep in vitro. So they are trying to find a way to reproduce these spindles. The spindles have a specific features in electrophysiology activity, and this is produced normally in non-REM sleep. And the literature say that this is because connection between cortex and thalamus. So what we are doing now is we generate thalamic organoids. We took this amazing protocol from Xiang and collaborators. So we spent some time first to basically characterize, so to make sure that the protocols work, because sometimes you see nice protocols in the literature, then you try and it doesn't work. So we spent some time, and we were lucky to see that this protocol works really well. And then when we were sure that we have a nice thalamic organoids with thalamic neurons, we moved to try to combine. So the idea was to combine these brain spheres or spheroids with the thalamic organoids, because our experience with some of the microarrays analysis that we have done is we have more cortex tissue in our model. So we thought that we could use that. And we tried different approaches. First, we try to combine the spheroids with the thalamic organoids. Unfortunately, the thalamic organoids that's why I was asking these days how people do it, because I am struggling to attach them to the well. And I think because our thalamic organoids have some bubbles in the interior or something, sometimes you see like a small bubble going in one of the side, and that makes it really difficult to attach them. So then we try to make them single cell suspensions of both. So we desegregate the thalamic organoids and the brain spheres, and then we put them together. And the other approach was to desegregate the thalamic organoids and make like kind of a carpet, like a monolayer of these cells, and then put the brain spheres on top and try to measure the electrical activity. So here just show some pictures of how it looks when we desegregate, because we also were worried that maybe if we desegregate the organoids, it's going to be a complete disaster, all the cells are going to die and so on. But Mochi have a really nice protocol, and we were able to obtain quite nice cultures after we desegregate them. I'm not really good with this pointer, I think. I don't know if it's better here. Well, here you can see this is a marker for thalamic neurons, and you can see how we get a lot of thalamic neurons, obviously with the thalamic organoid, what is good. We also have some markers to make sure that we also have like oligodendocytes, astrocytes, and so on. And here I put a picture of when we put this monolayer of thalamic neurons, and then we put the brain spheres on top. So yeah, so basically now what we are doing is to study by using mainly MaxOne, but now also MaxTwo, because that was what the lab was using before. And we are using this for not only for the iPS human-derived models, but also with mouse mouse primary cultures. And what we are starting seeing is when we combine the brain sphere with the thalamic organoids, we start to see these spindles. And I think we are close to finish the project. Now we are doing some single cell sequencing also to study, if the thalamic organoids are really representing thalamic tissue and these type of things. But hopefully this will be finished soon. And just to finish, I want to thank the people in my lab and my boss, Marie-Gabrielle Zurich, and also Meditafti group, and especially Moji, that is doing a lot of work on this sleep project. I want to thank also to Timothy Schaefer from EPA, because without him, it will be really hard to implement this into a regulatory purpose. Also, we have his knowledge about, all the chemicals we have to test and so on. But especially I want to thank MaxWell in general, because they are really nice and they have been really supportive of our work, but especially to Victor that was doing some of the analysis before, Anais has put a lot of effort lately into analyze the data, and Elena that is helping with these areas, development of the software, and especially to David, because he has to deal with me almost every week, and I know I'm a pain in the ass, so you are a superhero. So thank you very much. And now if you guys have questions, I'm happy to answer as good as I can.
And hello, I'm Kenta Simba of Jimbo Laboratory in the University of Tokyo, Japan. First of all, I'd like to thank MaxWell Biosystems for inviting me in this fantastic meeting. I'm very happy to be a speaker again. And I'm interested in the properties of the axon itself. We started using HD-MEAs to detect changes that occur at specific locations on the axons. Today, I'll talk about our recent study in axon conduction recorded with high-density MEAs from MaxWell Biosystems. Okay. So recently, some papers reported that axons modulate propagating signals. A study with HD-MEA has also reported daily changes in conduction velocity. Now, so axons are regarded as a computational unit in neurons. And here, I summarize some challenges in studying axon functions. The first one is the small size of axons. Therefore, conventional techniques requires high level of skills. When attempting to measure intracellular potentials with glass pipette electrodes, for example, it is necessary to drive a small patch pipette to attach the axon. And second, in measuring extracellular potentials, the axon signal is very small compared to cell soma. So researchers need to induce axon signal with a reproducible way and average multiple activities. So we have attempted to solve these problems by using electrodes combined with microtunnels. The tunnel structure allows axons to spontaneously grow on the electrodes, so no skill is required to place the electrodes on the axons. And the tunnel structure also measures high amplitude activity from axons, so eliminating the needs for special analysis method. So using this system, our alumni Dr. Sakai applied electrical stimulation to axon bundles and evaluated the changes in conduction velocity. The results showed that the presence of Schwann cells, a Schwann cell is myelin-forming glia, the Schwann cell increase the conduction velocity of axons even before the myelin sheath is formed on axons. So this result is very interesting, but this method also left some problems. The upper right photo shows axons and myelin sheaths in the microtunnels. Myelinated axons are very rare, and each myelin segment less than 100 micrometers long. Assume that a conventional MEA is used for this sample. Because the distance between the electrodes is more than 200 micrometers, so salutary conduction through one myelin segment cannot be measured. In addition, since electrical stimulation evoked the responses of multiple axons at the same time, the signals of a few myelinated axons would be masked by the signal of surrounding unmyelinated axons. So we began using MEAs with a higher electrode density from MaxWell Biosystems. And we believe that with sufficient electrode density, the conduction along specific myelin segments could be detected with high accuracy. Now we use MaxOne HD-MEAs for high spatial temporal electrical imaging. The right photo shows an immunofluorescent image of sensory neurons cultured on the electrode. The green color indicates axons, and red shows myelin sheath. The light gray squares on the right side show the size of electrodes. So we can apply multiple electrodes to a single axon or a single myelin segment, and we can record spatial characteristics along myelinated axons. To detect salutary conduction, we cultured sensory neurons and intrinsic Schwann cells from rat DRG, and we promoted the myelination by adding ascorbic acid. First, we confirmed myelination. When myelin sheaths are formed on axons, clusters of sodium ion channels are formed between two myelin segments, and adhesion protein, Casper, accumulates at the end of myelin sheath. So we stained the cell at 1 month in culture with some antibodies. The left panels show the result of sodium ion channels and myelin segment staining. The results showed the cluster of sodium ion channels between the myelin sheaths. And the right panel shows the localization of sodium ion channels and adhesion protein Casper. In many cases, the Casper accumulated on only one side of the sodium ion channels, suggesting that the myelin sheath was in the process of forming at 1 month in culture. Then we observed ultrastructure of the myelin sheath with electron microscopy. The blue color shows Schwann cells and the red color shows axons. In the first month, we observed a lot of unmyelinated axons, which are bundled by Schwann cells and developing myelin sheath. But compact myelin were rarely observed at 1 month. But at day 70, we observed a lot of completed myelin sheaths with more than 10 layers of myelin membranes wrapping an axon. And the parameters of myelinated axons are consistent with previous studies. These results indicate that the complete myelin is formed by day 70 in this culture condition. And we think if an ideal myelin sheath were formed, much of the axon would be insulated by the myelin sheath, and signals would be measured only at very small nodes of RAMVA, approximately 1 micrometers long. So under such conditions, it may be difficult to detect the axons as a continuous signal from the electrical activity. Thus, we performed numerical simulation to investigate the information processing required to detect axons. Parameters of myelinated axons were determined from results of immunostaining and electron microscope observations, and extracellular potentials were calculated from compartment model previously developed. The calculation results are shown on the right side when the distance of the electrode and axon is 5 micrometers. The upper right image shows the calculated extracellular potential and the position of the electrode. And the bottom right shows a signal measured at each electrode. Large signals are detected at the electrodes close to the node of RAMVA, while the signals at the other electrodes are very small. So the simulation results showed that it seemed difficult to detect saltatory conduction from the recorded signals without any signal processing. So we smoothed the two-dimensional images of the extracellular potentials for each time point. The figure on the right side shows the signal after smoothing and normalized by the maximum value. Smoothing makes the saltatory conduction appear to be one continuous signal. And now we think we can detect the axonal conduction from these myelinated axons. So we measured the axon conduction of rat sensory neurons. And normally, sensory neurons show little spontaneous activity, so we used optogenetic stimulation to induce activity with a blue light. The sensory neurons express the channel rhodopsin 2 and green fluorescent protein, GFP, after infection of AAV. These green cells on the left are GFP positive cells. Then we manually selected GFP positive cells and exposed blue light by DMD, digital mirror device. Here is a video of the experiment, in which each cell was stimulated individually and a single response was successfully evoked. By using optical stimulation instead of electrical stimulation, the stimulation artifacts are enough small and activity can be measured immediately after stimulation. Then, axonal signals were recorded from the entire electrodes in approximately 30 recording sets using the Axon recording assay included in the MaxLab Live software. Probably, Davide will explain how the Axon assay works. The data were then combined with respect to the spike time of the target cell, and the signal was reconstructed over the entire recording area. Each image was then smoothed. The movie shows the result. Red and blue color correspond to positive and negative extracellular potentials, respectively. The numbers on the right side indicate the time relative to the time when activity was observed in the cell body. And we can observe that action potentials propagated along axons. Then we detected negative peaks from the two-dimensional images, and the axonal conduction and the staining results were superimposed. The green color indicates the axon and cell body of the neuron, and red indicates the myelin sheath. And for clarity, the activity 1 frame before is shown in yellow and 2 frame before in white color. The distance from the red to white dots roughly correspond to the magnitude of the instantaneous conduction velocity. The axon potentials propagated mainly along the axon, with rotational changes in conduction velocity. These results indicate that with optical simulations, we can measure the axonal conduction from samples containing myelinated axons. Then representative axons were extracted and evaluated in detail to assess electrical features along the axons. Here are representative results: conduction delay, conduction velocity, signal amplitude, and electrical sink value. And the black scale bar indicates 100 micrometer. If a node of RAMVA and myelin sheaths are formed, inward current would be expected to flow only in the nodes because sodium ion channels would be concentrated in the node. So to evaluate the magnitude of inward current along the axon, we performed current source density analysis and calculated electrical sink, which indicates the magnitude of inward current. The axon was approximately 2 millimeters long, and action potentials propagate from the right side to the left. And in the lower left figure, we can observe high-speed segments with red or orange color alternate with low-speed short segment with blue color. In addition, larger amplitude and sink were observed at the low-speed segments. When salutary conduction occurs, large sinks are expected to occur in the nodes of RAMVA between myelin sheaths, so which is consistent with this result. In addition, a high-speed segment was superimposed on an immunofluorescent image for the myelin sheath. And in some cases, fast conduction occurred near P0 protein positive segments, indicated with red color in the left panel, suggesting that salutary conduction occurred. And the conduction velocity in the high-speed segments was around 2 meters per second, more than 5 times faster than the average of entire axons. And the amplitude ratio of the edge to the center of the high-speed segment was calculated to be greater than 1 in more than 19% of the segment. So this is consistent with the characteristics of myelinated axons. So overall, these results suggest that the HD-MEAs can measure the salutary conduction of myelinated axons. Up to now, I have showed our study for detecting salutary conduction. We think salutary conduction is a static characteristic because myelination takes more than 1 week. And now, as a next step, we are also trying to induce demyelination. And after demyelination treatment, we observed degeneration of the myelin sheath, as indicated by the black arrows in the photo. And the demyelination process also takes more than a few days. So these characteristics is not change quickly. And in addition to the static characteristics such as salutary conduction, dynamic properties are also important in the computational function of axons. So in the latter part, I will share preliminary results on activity-dependent changes in conduction properties. In order to evaluate the changes over a short period of time, we cannot use axon recording assay, which need to average multiple spikes. So we must select electrodes under the target axon in a single electrode configuration. So therefore, we first selected electrodes around the axon based on the result of the Axon recording assay. In addition, electrodes for electrical stimulation were placed under the cell body. In this setup, we measured the response to electrical stimulation using template matching to detect the spike from individual events. First, stimulus response at 0.5 Hz were used to create the waveform template. The response to 1 to 20 Hz was then detected by template matching. The movie on the right is a representative response to 0.5 Hz stimulation. The whole area shows electrical oscillation due to stimulation artifact, but within the artifact, there are some propagating signals. From the selected electrodes, we select one segment of an axon, showing in here, and we made waveform templates from electrodes in the red box. 9 electrodes. After increasing the sampling rate by spline interpolations, a cross correlation function between the template and each response was calculated, and the peak of waveform similarity was set as the spike time. The response time is shown in the lower right panel, indicating that the latency of response increased by 10 Hz and 20 Hz stimulation, suggesting that conduction velocity slowed down with repetitive firing. To further examine whether the decrease in conduction velocity occurred throughout the entire axon or at a specific part, the axon was divided into 5 segments, as shown on the left, and we calculated the conduction delay in each segment. When the conduction velocity was first calculated with a low-frequency stimulation, the 2 segments in the middle part had higher conduction velocity. Furthermore, plotting the response to 10 and 20 Hz showed that conduction time was extended only in the central red segment. This result indicates that the decrease in conduction velocity occurs in specific segment in axon. This segment is just where the axon is most crooked, and it would be interesting to see if there is a relationship with the shape of axons. Let me summarize my talk. We found that HD-MEA can be used to evaluate both the static property of salutator conduction and the dynamic property of activity-dependent change in conduction velocity. These results show that HD-MEA is an interesting tool in axonal research. And finally, I thank our lab member and collaborators. All experiments were performed in neuroengineering laboratory in University of Tokyo, discussing with Professor Jimbo and with lab members. Thank you very much for listening. I'd be happy to take your questions.